Aquaphotomics prototypes.
Jelena, what is the second, the other one? Let's move to another.
Sorry, Felix, thank you so much. This was really nice.
Felix, yes.
Stay in touch regarding this. We have 2 more presenters, and maybe it is nice not to go further now, because if we go further to Ponton, we're going to end up around midnight. Let's go to more precise topics like food quality, and we have Zsanett Bodor and John Louis Ziniazaku. So who is ready from the 2 of you? Zsanett?
I can start.
Ready?
Yeah.
So, you have very, very nice topics, and you are, I think, joining the avalanche of the people who are investigating adulteration of honey. So that's very nice. And what can you tell us what is the common conclusion? What it means to adulterate honey in terms of water?
Can you see my screen?
Yes.
Yeah, okay. So good afternoon, everyone, and good evening, everyone in Japan. So I'm Zsanett Bodor, and I'm working in Zoltán's research group, and I'm working with honeys for 6 years now. And when Zoltán brought up the idea to try the aquaphotomics in honey, I was really interested, and we decided to do some experiments, I think 2 years ago, on the application of aquaphotomics to reveal the adulteration of honey. And maybe not all of you are familiar with it, but honey is really a big target of adulteration practices. And from these, one of the most common one is the direct adulteration, which means that the honey is mixed with different sugar syrups. And we have done several pre-tests before in higher concentrations. So we have adulterated honeys up to 50%. But now we decided to do studies regarding smaller concentrations. So what I brought here today, it aims to determine the potential of aquaphotomics to detect and quantify the adulteration of honey at lower levels. And in this experiment, we have adulterated honey with rice syrup and corn syrup, which were from commercial origin. And I also created a sugar syrup from fructose and glucose, and to have about 20% of moisture content, which is really similar to honey. And we have done all the samples with 3, 5, and 10% of adulteration. We did every sample in 3 repeats, and we applied the benchtop instrument to get the spectra, and each sample was measured with 3 consecutive scans. And for the data analysis, we used the first overtone region in R using the AquaP2 software. And what I wanted to highlight regarding the results, that it is really obvious based on all the models that we have built based on PCA, LDA, or PLSR, that one of the most important conclusions that this applicability is really dependent on the type of the syrup what we add to the honey. And I'm sure about that even within syrup, so for example, different sugar syrups from rice syrup are also would give some different changes. That's why it's really important to do more and more samples and analyze them and build a big database to be able to make really good conclusions. But we could see that the self-made syrup was more hard to detect. In my case, so I'm a really good adulteration people, so I can do it very well. Yeah, and the syrups from commercial origin was more detectable. In the case of the LDA, we could see that in the case of the rice syrup, we obtained 100% accuracy, while in the case of the self-made syrup, it was lower. And the PLSR results were also a bit different for the 3 types of syrups. But what is really important for us from aquaphotomics point of view is that we did an aquagram using the 3 repeats of the different syrups and the control honey. And what you can see that the self-made syrup structure is really more similar to the honey structure than the commercial syrups. That's why maybe it was really more hard to detect this sample, and this kind of syrup were more characterized by and the honey with strongly bounded water, while the free water was higher in the case of the rice and the fruit, so the corn syrup. And if we go into details regarding the aquagrams of the different syrup adulterations, we can see that the trend is also nicer in the case of the commercial syrups. So we can see that if we add the syrup in higher concentrations, then we will have more less bounded water in the samples. And this is what we wanted to highlight also in the results, that first of all, you have to be really careful about the syrup types. So you have to determine more and more types, more and more honey types also, to have a big conclusion. And that that usually this kind of commercial origin honey, and it is true from our previous studies also, that they are more related with the free water, and while honey is usually more related to strongly bounded water. And I think it was also mentioned in George's paper before. So this is what my topic is about.
Thank you, Zsanett. So when I see something like that in my own results, I usually have to turn my face to the back of the lab and say, oh, I'm in love. Because your poster presents so many beautiful things. First of all, when you look at the aquagrams, it doesn't matter what happens. Adulteration of honey with syrup is changing the face of the honey, and that is exactly, for example, something that you can see in the temperature papers. For example, as the temperature of the water is cooling, you will have the same pattern. And you had very, very interesting band, which is different for different adulterants. Something about, I think I saw 3 different levels at 1440 for this. So, and that band, for example, was found to be important in this resurrection plant, Cabernet rhodopensis. So, phase transition is important, something for the preservation of tissues. And as I noticed that you are from dietitian, sorry, I don't know how to say the word. This seems to be very, very important for the basics of the health. The phase transition of water. I don't want to talk more, I talk too much. So we have a question from Pierre. Pierre, can you see the question in the chat?
Yes, yes. So he's asking about okay. I'm not sure I understand the question. So yeah, of course, sugar is not only sugar, and in the case, of course, we are talking about a syrup, to say so, which means that it also contains some kind of sugars, water. And so it is also a liquid form, and this is why they usually add it to the samples, because it is really similar in, how to say, so in looking. So it looks like honey sometimes, and it's really hard to detect by eyes, and that's why they can use it to fake the honeys, because it is really similar. And just to tell some information about this, even with a really, really precise, I don't know, LC-MS or mass spectrometry results, these syrups are really, really hard to detect. So that's why we do this study, to have this, how to say, to build a model and a method which is really easy to use, and to find if it's applicable, because there is no method for this since now.
I'm glad you mentioned the work of George, and of course you're aware of this, and kind of he started this. what Pierre is saying is also very important. Today we were talking about biocompatibility in material science, and what he's pointing out is that honey is the most biocompatible sugar, and you show it by honey's aquagram. And I just want to mention one thing that I was thinking while listening to you. So we had this, I think, with the council also. I think the first thing that you need to do is to make a big database of the most beautiful honey on earth, the most real honey. They have different colors, they come from spring, from summer, autumn, different locations, but they should have one specific aquagram, all of them. I mean, and that aquagram should describe the biocompatibility of sugar. A very good point, very interesting. And then what Jelena just mentioned, the phase transition and everything added to this will be fake. It will be very easy to identify. So yeah, I think this is the way to go.
Actually, this is our further plan, because just to tell you, I'm now in Germany, the AD scholarship, and now I will go back to Hungary. We are planning to do a very, very, very big experiment regarding this, where actually our aim will be to use more types of honey from Hungary, and from one type also honeys from different origins, because also the geographical origin, for example, in chestnut honey, if it's from the eastern part of country, eastern part of Hungary or the northern part of Hungary, they are really different. So our aim is to build this database, and actually we have already measured, I think, 200 different honeys from different botanical origins, but it's another question. So now we are going to do a very big experiment. I don't know, really large samples. I mean, this is the plan, to be able to really build this database that you were talking about, to be able to build models that are really, really trustable. And so this is the
very important knowledge for the whole world. So we can send you some samples from Japan of real honey. Yes. No, it is I think very, very good. And Hungary exports lots of honey. It is very important for Hungary, right?
Yes, about 80% of our honey is exported. Yeah, thank you, Pierre. It would be really nice.
Yeah, I just wanted to say, and you got a present from Sicily.
May I ask something?
Yes,
sure.
In our university, now we have a project connected with the quality of honey. We have some samples. It's possible to exchange.
This is Bulgaria. It's Bulgaria.
This would be really good, I think.
University from Bulgaria.
University of Trachea. Trachea University in Bulgaria.
Also, our colleague prepares some special honey. They feed with some sugar solution. They feed and they have some kind of honey.
I think that we can talk about this exchange later, or maybe have a meeting on it in the future and discuss about the possibilities how to do this, if it's good for you.
Well, you have many offers, obviously.
And just one word to Petya. Yeah, it's a really nice idea, and actually it would really worth to analyze. But the problem that in Hungary these GM plants are not really allowed to plant, if I know well. Therefore, in Hungary it would not be possible, but maybe from other origins we could do this. But yeah, it's a big question. And I have read. Sorry?
From Spain. I think in Spain they allow genetically modified.
Yeah. So yeah, maybe that would be a good collaboration with someone from there. This would be very nice. Thank you for the ideas and for everything.
Thank you, Zsanett.
Thank you.
I'm sorry, Zoltan, you wanted something, maybe? No, I'm wrong.
I just agreed, and
you agreed.
And thanks for all the offers. We are expecting a better spectrophotometer if everything goes well, and then we can collect even more stable spectra. And I think this will be when the real game will start, related to building up a database and going deep down in the water matrix coordinates as well. So thank you for all your kind offers and feedbacks.
Good luck.
Thank you very much.
Good luck, Zoltan.
Thank you, Zsanett. Thank you very much.
Thank you very much.
And we have one more person who has the honor, I think, to finish this day, if he's still alive. John Lewis, you there? John Lewis.
Yeah, I'm here.
Hi. Hello. Thank you so much. You have been very patient.
Thank you so much.
Can you show us your poster? You have a neat topic. Food related, very practical, and I think Professor Atanasova might have questions and comments. This is really her topic.
Just a minute.